The ethical considerations of gene modification. For thousands of years, humans have felt the need to understand the world around them and eventually manipulate it to best serve their requirements. There are always ethical questions to address, especially when the manipulation involves the mortal genome. There's currently an urgent need to assiduously pursue those conversations as commercial gene sequencing and editing technologies have become more accessible and affordable. This video explores the ethical considerations of gene editing and hereditary selection, including the hurdles scientists will face in trying to develop new technologies into feasible remedial options. We're glad you're here. Are you ready to hear about the most incredible future applications of CRISPR gene editing technology? Let's examine how CRISPR might influence the future by curing diseases and extending the human lifespan. In this video, we will show you how using CRISPR might cure diseases and extend our lifespan, as well as the ethical considerations of gene modification. In 1987, a laboratory in Osaka accidentally exposed an unusual palindromic repeat sequence in the E. coli genome they were studying, exceptional in that it was regularly interspaced. These DNA motifs were further recognized in various bacterial genomes by multiple laboratories over the next 20 years. Their function, however, was still unknown. By 2005, 3 groups had independently determined that the spacer sequences were actually derived from phage DNA, and the probability of the genes playing a role in bacterial immunity was first suggested. By this time, the scientific community referred to this unusual array as clustered regularly interspaced short palindromic repeats, or CRISPR. Meanwhile, researchers in the Netherlands had identified some other genes located near the CRISPR locus that appeared to be functionally linked with the CRISPR genes. 34 of these would turn out to be the CRISPR-Cas-associated proteins that make up an integral part of the CRISPR-Cas system. In 2007, the CRISPR-Cas system was identified as being a prokaryotic defense against pathogens as part of a self-non-self determination contrivance of adaptive immunity. Prokaryotes integrate a segment, usually 32 to 38 base pairs of phage DNA, into their own genome, generating the spacers in the CRISPR arrays. After the CRISPR genes are transcribed, endoribonucleases cleave the resulting CRISPR RNA, pre-CR-RNA, resulting in shorter RNA units composed of a single spacer sequence and the palindromic repeat CR-RNA. Contingent on the organism, a transactivating CR-RNA, tracker RNA, may also be transcribed. The RNA forms a ribonucleoprotein (RNP) complex with the associated Cas proteins. Any phage DNA containing the spacer sequence will be identified by the guiding RNA and rejected by the endonuclease function of the Cas proteins. The protospacer is the homologous sequence in the invading DNA and is followed by a short protospacer adjacent motif. Because the PM is not incorporated in the CRISPR array, the CRISPR-Cas complex is able to distinguish the foreign DNA as non-self and thus will not cleave the prokaryotic cell's own DNA. In 2012, Jennifer Doudna, Emmanuel Charpentier, and others on their team engineered a synthetic chimera of the tracker RNA and CR-RNA, now known as a single-guide RNA or sgRNA, which was able to direct the Cas9 to create a targeted, site-specific double-stranded break. By 2013, investigators had established that the CRISPR-Cas9 was an actual facetious and multiplexable method of editing the human genome. Newer CRISPR-based editing methods do not rely on fickle NHEJ or donor DNA. For example, endonuclease-deficient Cas proteins can be fused to base editing enzymes, first described in 2016. Researchers have recently reported a high-fidelity base editor with no off-target mutations, OTMs. Epigenetic techniques are also being reconnoitered using CRISPR-Cas technology, including linking endonuclease-deficient Cas proteins to effector molecules, and prime editing reports genetic disorders caused by multi-base variances such as sickle cell and Tay-Sachs. In this case, the impaired Cas9 is fused to an engineered reverse transcriptase. ZFNs and talons do sustain some advantages. CRISPR entails a PM sequence, and sgRNA spacer sequences are usually only about 20 base pairs, meaning an inherently reduced targeting capacity. The researchers have recently begun exploring the effects of increased sgRNA length on cleavage efficiency and target specificity. CRISPR vectors are also necessarily larger, making delivery more difficult. Overall, however, CRISPR is usually the preferred method of genetic and epigenetic manipulation, especially as improvements are made to the technology. CRISPR's main benefit over its predecessors lies in the fact that rather than a complex protein as the DNA recognition molecule, the CRISPR system relies on a guide RNA. CRISPR kits are thus notably cheaper, easier, and more competently produced than either ZFNs or TALENs. Ethics of gene editing. On November 25, 2018, news broke that He Jiankui of the Southern University of Science and Technology in Shenzhen, China, had enumerated a clinical trial in which he planned to implant human embryos which had been reformed using CRISPR-Cas9-64. Within days, the world learned that not only had edited embryos been implanted, but 2 baby girls, Lulu and Nana, had already been born. He used CRISPR-Cas9 to create a nonspecific sequence alteration in the CCR5 gene. CCR5 is a 7-transmembrane-spanning G-protein-coupled CC-chemokine beta-chemokine receptor. When articulated on the surface of a human T cell, CCR5 is the main coreceptor along with CD4. For the human immunodeficiency virus, HIV, a naturally occurring 32 base pair deletion with heterozygote allele frequencies of about 10% in people with European origin, known as CCR5 delta 32, has been shown to incapacitate the protein. 66 heterozygosity of CCR5 delta 32 allele has been shown to slow disease progression, while homozygosity ominously increases disease resistance. He's goal was to knock out CCR5 with the desired outcome of creating HIV-resistant babies. It should be noted that HIV infection in CCR5 delta 32 individuals has been increasingly reported associated with X4-tropic HIV strains, that is, strains that rely entirely on coreceptor CXCR4 for endocytosis rather than CCR5. He presented the details of his investigation at the Second International Summit on Human Genome Editing, being held to discuss scientific, medical, ethical, and governance issues associated with recent advances in human gene editing research. While his manuscript detailing the trials was not accepted by any publications, excerpts are available to the public, and several media outlets and some experts have been able to view the paper and supplementary data in their entirety. Enough is known about He's work that it can serve as the basis of a conversation about the ethics of joining germline gene editing. There are a number of issues, those inherent in the technologies themselves, as well as scientific hurdles, that need to be overcome before starting clinical trials to ensure that they are carried out as ethically as possible. That's all for today. Leave your thoughts and comments in the section below, and don't forget to like, share, subscribe, and turn on the bell notifications so you don't miss any future videos.